So as I mentioned, we were going to talk about tonicity. And tonicity is going to be the ability of a solution to change shape or the plasma membrane tension, we call it, of cells, all right, by altering the cell's internal water volume. All right, so in short, this just means the ability of the red blood cell or the cell to expand or shrink. Okay, now isotonic, iso means same, tone or tonicity. Solutions that are going to have the same concentration of these non-penetrating solutes, that means that they can't move back and forth across the cell, are going to be found in cells. So this is going to be the same concentration as your cells. So the 0.9% saline, we call it 0.9 or 9% sodium chloride, or 5% glucose. This is basically what we would consider isotonic, because it's the same as what you would see in the cells. So you're not going to have any net movement of water in or out of that red blood cell. All right, so this kind of just goes over when we expose a cell, a red blood cell, to isotonic solution, which means that it's the same tonicity on one side of the plasma membrane than the other. All right, these blood cells are going to retain their normal shape. They're not going to expand or they're not going to shrink, and you're going to have no net loss or gain of water. Okay, so water may move back and forth across this membrane, but it's not going to result in a gain or a loss of water. Okay, it's just going to stay the same. Now, most of the body's extracellular fluid and most IV solutions are considered isotonic. So remember that iso means same tonicity, and you're not going to have any change in the plasma or the cell when that substance crosses the plasma membrane.